Assalamu alaikum. We have already started the vectors for eukaryotes in our previous lecture, and today we are going to continue the same. And our learning objectives for today's lecture are: which vectors we use for cloning in higher plants, what are agrobacterium plasmid based vectors, what is meant by direct gene transfer in plants, and which type of viral vectors can be used for plants. Okay. Now we need to understand now that why do we need to clone genes in plants. So obviously we have certain aims. That's why we are looking forward to have plants as host. The obvious aim is to make transgenic plants so that we can get our protein of interest expressed in the plants. Or secondly, we want to use plants as factories to produce our protein of interest, or to improve certain traits into the in the plants, like flavor, like color, and like to increase the productivity, to create the resistance against the pathogens, so on and so forth. Okay, so cloning vectors for higher plants were developed in 1980s, and that led to the genetically modified GM crops. Now there are three types of vector systems which are used for plants based on the naturally occurring plasmids of agrobacterium, using the direct gene transfer, and we can transfer various types of plasmid DNA into the plants, and based on the plant viruses that will naturally take our gene of interest into the plants. Now, vectors based on agrobacterium. So agrobacterium is a very important bacterium which is living in the roots, which is living in the soil around the roots, and whenever it sees a crack or wound in the plant root or the stem area, it will try to dwell in, and as soon as it enters, it tries to create a niche for itself and results in the formation of a cancerous appearance or a tumor-like appearance on the plant, and that is called a gall. Okay. Now, agrobacterium is also termed as nature's smallest genetic engineer. Why it is called smallest genetic engineer? Because where it enters into the plant, those plant cells it genetically modifies to make its own food. Okay. Now, how does this bacterium is attracted towards plant? Obviously, when there is a wound or crack in the plant, so there are certain compounds they are coming out of the plant, and they are attracting this bacteria towards this site where it goes, and mostly it infects the dicot plants. Okay. During the infection, a part of a plasmid which is known as the TI or tumor inducing plasmid present in agrobacterium is integrated into the plant chromosomal DNA. That's why it is termed as the nature's smallest genetic engineer. Now let's just have a look how do agrobacterium infect the plant. So agrobacterium, here we see agrobacterium containing a TI plasmid. This one is the agrobacterium's own genomic DNA. The TI plasmid contains the valence genes, okay, and it contains a portion known as tDNA. Once a plant is wounded, so phenolic substances are released from the plant, and these phenolic substances they attract the agrobacterium to come and attach to the surface of the plant cell. Okay. Now, Vir-A and Vir-G proteins which are present here in the agrobacterium, they activate the valence genes, and this causes the agrobacterium to prepare itself to enter its tDNA inside the plant cell. Okay. Now, other Vir genes encoded protein function in tDNA transfer and tDNA nicks from the right border. Nicks mean a small nick is created, okay, and as a result the tDNA nicks from left border and then enters into the plant cell. Okay. Alright, now tDNA, after it enters a plant cell, it integrates into the host genome. It can be seen clearly over here. And these two pictures, they are taken from Ozan et al. 2004. So you can the reference is given here. Now you can see here, this is Agrobacterium tumefaciens having a Ti plasmid. The Ti plasmid has tDNA, and then what causes the tDNA to move in? Obviously the phenolic substances, they attract the bacteria toward the plant cell, and when the bacteria attaches itself to the plant cell, the villous protein present inside the bacteria, they activate the Vir genes, and the Vir genes then prepare for transfer of this tDNA inside the host genome, and as a result the host genome is modified in the and this does not result the whole host plant to become a transgenic. Only the localized area, the cells become transgenic. Now let's just have a closer look at the tDNA and the Ti plasmid. The Ti plasmid, also known as tumor-inducing plasmid, it is more than 200 kb in size, and after the infection, one part of it, the tDNA, it integrates into the plant chromosomal DNA. This is called tDNA because this is actually the one which is related to induction of a tumor. Okay, the size of tDNA is 15 to 30 kb, and it is maintained in a stable form and passed on to daughter cells as integral part of the chromosome. The tDNA contains 8 genes that are expressed in plant cell and are responsible for cancerous properties of transform cells. So this figure I have taken from the book T.A. Brown and chapter 7. Now let's have a closer look on the Ti plasmid, and this I have taken again from the Naureen 2002 and Ozan 2004. So here the tDNA has a left border, has a right border, and it contains genes which are making plant hormones like auxins and cytokinins. Okay, and here it is also having gene for synthesis of opines. These opines are used as food by the bacterium Agrobacterium. Now the reason why the plant cells at the site of infection become cancerous is this: auxins and cytokinins. These auxins and cytokinins both are secreted in a proportion which leads to the plant cell to become cancerous. They will start to grow and they will not be differentiated into any organ. Okay, that's why they lead to a tumorous growth formation or cancerous growth formation. Okay, now here there are several Vir genes in the Villoins region. So Vir H host range it specifies the host range, and Vir F nuclear targeting of single-stranded DNA, and Vir E single-stranded DNA binding protein, and then this is coding. Vir D is coding for an endonuclease, and Vir C is overdrive binding protein. Vir G is a Vir activator which activates this region, and Vir B is a membrane protein, and Vir A is a wound signal receptor. Besides that, there is a origin of replication and incompatibility region, and that's why you see this tDNA or Ti plasmid cannot be transferred to the monocots. Why? Because it is specific for the host. Here we have TRA genes, which are transfer gene, and they are actually required when the bacteria is having conjugation with other bacteria. And octopene catabolism genes, they are also present here. Many-tyl-opene catabolism gene, which are some metabolic gene involved in the metabolism of the Agrobacterium tumefaciens, they are present here. Then, how can this Ti plasmid be used as a vector? So, one of the main problems for converting this Ti plasmid into a vector is its size. Obviously, the size is too big, and when the size is too big, it leads to another important problem, and that is unique restriction enzyme site. Because the size is too big, so unique restriction enzyme site is not present. Okay. So what is the solution then? So there are two methods which scientists have used. One is the binary vector strategy and other is the co-integration strategy. Okay. And obviously the reason of using TI plasmid as a vector is to insert the gene into the tDNA so that this tDNA is integrated into the plant chromosomal DNA and leads to the formation of a transgenic plant. Okay, let's have a look at the binary vector system. Binary means two, so having two vectors. So as the figure 7.11 taken from the T.A. Brown gene cloning and DNA analysis, T.A. Brown chapter number 7. Here we see plasmid A. Plasmid A contains only the valence region and the host specific region, while the plasmid B contains tDNA. Now this is the tDNA which we want to be transferred into the plant host, while the tDNA cannot move by itself unless until it has valence region and the host specificity region. Now valence region, all the genes are present on plasmid A, so the plasmid A actually will help the transfer of tDNA from the plasmid B into the host chromosome. Okay. Right. So a more closer look over here. This is a binary vector and this is a Weir helper TI plasmid. The Weir helper TI plasmid, it is same as this one plasmid A and it contains valence region and host specificity region. While the binary vector, it contains the region of replication, a left and right border for the tDNA, several multiple cloning site, and it contains the selective plant marker gene and antibiotic resistant gene and origin of replication and the transfer region. Okay, and RK2 is another replication region which belong to the plasmid. Okay, so this is the overall binary vector which is used in association with a helper plasmid. Now the second is co-integration strategy. The co-integration strategy aims at integrating a small tDNA after cloning the gene of interest in it with the bigger TI, the normal TI plasmid. So, so what is the basic aim of co-integration strategy? The co-integration strategy has small portion of tDNA, mostly the left and right border, and it has multiple cloning site where we can add our gene of interest. Why the size is kept small? The size is kept small so that we can have unique restriction enzyme sites and we can easily manipulate it. After cloning into this small E. coli type plasmid, we let it to have a homologous recombination with the normal TI plasmid, and as a result, this whole small plasmid integrates into the tDNA. Now we have a defective tDNA which contains our gene of interest. Now this defective tDNA will not be able to cause any disease into the plant, and this recombinant plasmid will be able to transfer our gene of interest into the host plant. Here we have taken a co-integrate plasmid and its component, and this I have taken again from Ozan 2004. Okay, so let's just have a look at this. It has a left border, it has a right border, it has origin of replication for the transfer. It has called E1, which is replication origin in the bacteria, and then restriction, antibiotic resistant gene, then the plant selectable marker, the multiple cloning site, and almost similar to the binary vector we saw in the previous slide. Okay, now how do we transform the plant using the TI plasmid? So we have just studied previously that when we apply the recombinant bacteria onto crack and wounds, then what will happen? It will cause a localized transformation of plant cells. Okay, so clone gene will only be present at the site of infection. So how to overcome this problem? So to overcome this problem, what do scientists do? They first do the plant tissue culture to get the plant cells. So by using the plant tissue culture method, we first get a cell suspension culture of the plant which we want to transform, and then we incubate the plant cell suspension with the recombinant agrobacterium tumefaciens. Now, the recombinant agrobacterium tumefaciens passes on its tDNA into the plant chromosome, and as a result, the plant cell becomes transformed. Now, these are plated onto a solid medium, a selectable, which contains selectable substance, for example, any antibiotic resistant gene or any selectable marker can be used over here, and this results only the transformed cells will develop into calla, okay, into the callus. Callus is an undifferentiated mass of cells, and obviously this media will contain equal proportion of the auxins and cytokinin, and that will let this cell to develop into an undifferentiated mass of cell rather than developing into a plant organ. Then from here we transfer these calli, we subculture them onto a media which has a different hormone balance so that we can develop shoots. After that, we let the plant to grow in soil. We just put them onto a rooting medium, and then we let the roots to develop, and then we can grow our transformed plant into soil. Okay, this figure also is taken from the gene cloning and DNA analysis. In this figure, we are going to see the practical application of agrobacterium mediated gene transfer to develop transgenic cotton. So this figure again I have taken from a paper, and the reference is given here. So agrobacterium mediated gene transfer protocol for cotton. So first of all, in A, here we are doing the surface sterilization of the seeds. Then after surface sterilization, the seeds are planted, and 1 week old plantlets are taken, and the meristematic part, the actively growing part, is taken. Okay, it is cut. Now this meristematic part is then grown on the plant tissue culture media, and the plant tissue culture media, here we develop it into callus, and then subsequently we get the plant cells. Okay, now here the plant cells, they are then transformed using the agrobacterium tumefaciens. After transformation, they are grown again on plant tissue culture media and containing the antibiotic kanamycin. Now, because we have kanamycin resistant gene in our plasmid, so only those plant cells will be able to grow and make calli which have taken the tDNA. Okay. Now later on, the transformed cells are grown separately, and they are used to get the transgenic plants, same as we saw in the previous slide. And then the transgenic plants, they are checked for the presence of the gene using the histochemical and molecular analysis, and then the selected transgenic plants are then put on the shooting and rooting media, and then they can be potted into soil. Now, similar to agrobacterium tumefaciens, there is another bacterial species, the agrobacterium rhizogens. The agrobacterium rhizogens is a rod-shaped gram-negative bacteria. It is a non-spore forming bacteria, and also it develops in the soil. Agrobacterium rhizogens, they are characterized by the presence of a large root-inducing Ri plasmid. Like we studied the Ti plasmid, the agrobacterium rhizogens, it has the Ri plasmid. Okay, and it contains a highly conserved core DNA region required for a condition called hairy root. Now, agrobacterium rhizogen, it also infects the plant but in the plant roots, and it causes extensive growth of hair. You can see here root hairs on the root. So that is called hairy root disease. Now let's just have a closer look at the root inducing RI plasmid. It is again a very large plasmid, 250 kb. It has two tDNA fragments, the tRDNA and tLDNA, the right tDNA and the left tDNA, and this is separated by a 15 kb segment. The tRDNA is similar to the tDNA. It has genes for auxins and agropine synthesis. And agropine is again a type of opine or opine. Now tLDNA has 4 genes, role A, B, C and D, and it is for the root loci. So where exactly it is going to infect the root, this is determined by the TL region. Then RI plasmid has a villus region that mediate the transfer of tDNA and an origin of replication same as the TI plasmid. What is the use of this RI plasmid? The RI plasmid can be used as substitute for TI plasmid to transfer foreign gene into the plants. And RI plasmid can also exchange DNA segments between plants and bacteria, and hence they could be used to study gene exchange between the organisms. They can be used to induce rooting in the clones where rooting from calyx or explant is really difficult. Okay, like for example, if we are, they can be used in the plant tissue culture for those plants where we cannot induce rooting. Okay, they could also be used to increase nodulation frequency of plantlets during the micropropagation. There are two main types of RI plasmid, the monopine type here, and the agropine type. What is the difference? Let's just have a look. So the tDNA in both of these has a difference, that's why they are different. If you look at other regions, the Vir region is same here and here, then the transfer or conjugal region is same, then a vegetative origin of replication is same, and opine catabolism gene is same, and as well as the octopine catabolism. But if you look at the tDNA in both, there is a difference. The role A, B, C, D genes are same on the left side, while if we look at the right side here, so there are different genes and there are different genes. Okay, here we have auxin genes also, which we cannot see over here. That's why we have two different types of RI plasmid based on the presence of genes in their tDNA. Okay, now here the tDNA is single tDNA, while here the tDNA can be split into two regions. Okay, here you have a left border, here you have a right border, while here again this can be differentiated into the left tDNA region and the right tDNA region, as we discussed in the previous slide. Okay, let's just have a look at how RI plasmid is used for transformation of the plants. So like TI plasmid, RI plasmid also can be used to develop a transgenic plant for the production of the secondary metabolites and for the gene function analysis in plants. So unlike the Agrobacterium tumefaciens, the Agrobacterium rhizogens, it is causing the hairy root disease. So what we will do here on the plants, we are going to make the hairy root. Okay, now how do we do it? First we grow the Agrobacterium rhizogens onto culture media, then we incubate the plants which have wounds on their surfaces made by the sterilized needle, and we incubate these plants with the Agrobacterium rhizogens. Then Agrobacterium rhizogen at the site of wound, it starts to induce the hairy roots. After we develop the hairy root culture, we can use this hairy root culture in several ways to make the transgenic plants. Okay, now before moving on to the use of this hairy root culture to get the transgenic plant, we confirm the transformation of the plant by molecular methods. Okay. Now after that it is confirmed that the root culture is transformed, then we can develop the callus from the root culture. Okay. And this callus can be used to regenerate the shoot and develop a transgenic plant. On the other hand, the hairy root culture can be used to make encapsulated beads, and what we can do is we can cut these root section into small sections, and they can be incorporated into the alginate beads, and they can be used as seed to develop hairy root cultures, and then we can make the shoot. Okay. Now in another way, what we can do, we can grow these hairy root in liquid media, and we can use these hairy roots as a factory to make our protein of interest at a larger scale. Okay. So transgenic root systems are useful for introduction of new genes and tDNA, and this is used for altering the metabolic pathway and production of our compounds of interest, like the pharmaceutically important metabolites. Okay. Now there are several advantages of agrobacterium mediated gene transfer into the plant, and there is a big disadvantage as well. So looking at the advantages first, it is a very simple system, easy to set up and use, and it requires minimal equipment. Okay, and after that, the gene which we have cloned into the plant using Agrobacterium tumefaciens is actually having a high stable transformation. Okay, and minimal DNA rearrangement, so there is no DNA shuffling that can destroy our gene of interest. But the big disadvantage is that Agrobacterium tumefaciens cannot infect monocots, legumes, and woody plants. And most of the plants, most of our food crops, they are monocots and legumes. So for this reason, we have to use other methods of gene transfer into the plant, and these other methods include the viral mediated gene transfer and the direct gene transfer, which we are going to study in our next lecture. You can have. So here are some references which I used to prepare this lecture. You can have more details of what we studied today by referring to these references. Thank you.